some drugs need to be done clinical, but you can get a bio waiver if they are qualitatively and quantitatively similar. But this is easier said than done, and most studies do end up conducting a clinical endpoint study. Those in formulation would agree that it's very difficult for creams and ointments to get similar Q1 and Q2. So to sum it up, there are different topical products available. These are some examples of how different approaches need to be applied based on the action of the drug, its absorption into the bloodstream, and the requirements of the regulatory bodies. Some examples for the clinical endpoints. So for dermatology, it's the number of acne lesions before and after treatment, is the psoriasis index for the psoriasis studies, clearance of actinic keratosis. These are the endpoints for the derma studies. In the respiratory studies, it is the difference in the FEV1s, or some of the studies require a provocative study, that's a methacholine challenge test to be given to prove bioequivalence. Gastrointestinal, there are again clinical endpoints like the complete spontaneous bowel movement for linaclotide, or straining at stools as the endpoint. Ophthalmic, the intraocular pressure for glaucoma drugs, or the number of inflammatory cells at the end of the cataract operation. Otic studies for otitis externa and media are again topical studies. Vaginal estradiol for the symptoms of vaginal atrophy is again a local study with a clinical endpoint. Main challenges with the study design of the clinical endpoint. Most of these studies are a 3-arm study with a test reference and a placebo. They're double-blind studies because these endpoints are usually subjective, and the investigator should not have a knowledge of whether it's test or reference. It's a parallel design. It's a patient population against the healthy volunteer population. The sample size is difficult to determine because you can't do pilots in these kind of studies just like you would do in the PK studies. And again, there is a high variability due to the disease factor. The concomitant medications control is very difficult, whereas in a bioequivalence study, you would want absolutely no concomitant medications to get a true difference of the two drugs. The role of placebo in these studies is highly discussed, and it's a challenge when presented to the ethics committee. It's a challenge when you present it to the DCGI for approval. The scientific explanation for a placebo is that it is required because the test and reference should show equivalence, and each of them, that is the test and reference, should be superior to the placebo. So the placebo arm is recommended to demonstrate that the test and reference are active. So one is that the products are active, and it also establishes that the study is sensitively designed to detect the differences between the products. You would agree that some of the endpoints like acne lesions, let's take a very simple one. Acne there can be a placebo effect. It does get resolved on its own, and there, if it's just the test and reference, probably they may show equivalence, but the study may not be sensitive enough to detect the differences. And therefore, placebo arm does ensure that the study is sensitive. So when you are able to demonstrate that test and reference are superior to the placebo, that means your study is well designed, and then the next step is to show the equivalence between the two.